This is podcast number 36. Okay, in the last podcast, we were talking about myelinated neurons. Myelinated neurons are created by Schwann cells, and the myelin is a special membrane wrapping. So the cell wraps around the cell, the axon of a neuron multiple times. Axons and Schwann cells are different cells that work together. And then we talked about the analogy of why it is that myelinated cells can conduct action potentials faster, and that's because without that myelin, you lose the signal along the way because you lose those sodium ions due to leakage, like this, because they aren't prevented from leaking by myelin. So taking that into account, here's an H5P question for you. There are different kinds of synapses. So a synapse is connection from one neuron to another neuron. If the action potential is conducted all the way down that neuron, then you want it to connect to another neuron to get to, say, the ultimate target, which may be an organ, maybe the brain, maybe muscle, whatever. So at the connection between cells, the connection can be electrical in nature, in that there is a physical connection between the cells made up of a channel formed by multi-subunit proteins. And as this presynaptic cell depolarizes its membrane, these are voltage sensitive, so they're voltage gated, these connection units for this protein. And they change shape in response to the change in voltage that arrives at that part when the action potential arrives. And because they change shape and they're physically connected to proteins in another cell, the next cell, and they will physically change shape and cause the propagation of the action potential in the postsynaptic cell. So this voltage change is detected in this membrane, causing these proteins to change shape. These proteins change shape as a result because they're physically connected. That causes a voltage change in the postsynaptic cell, and the nerve impulse is conducted from one cell to the next. So that happens very, very fast. But in terms of the quality of the signal, there's not much variety possible because it's just on or off. In contrast, chemical synapses, which are more common, and they come with some variety because of the different properties of the chemicals that can transmit that signal. So these are typically drawn as a space in between two cells called the synaptic cleft, and that's exaggerated here just so that you know that there's a physical space by which these chemicals have to cross. But in reality, that physical space between one cell and another cell, you can see it on an electron micrograph, but it's small. It's a short distance. So in these sorts of situations, again, you have an action potential traveling down a myelinated or unmyelinated neuron, and that will change the membrane voltage that is detected by proteins at the synaptic cleft, at the postsynaptic or the presynaptic terminal. So this is where the synapse is going to happen, and that will cause synaptic vesicles, these little balls here that are filled with neurotransmitter, to come to the membrane surface and fuse with the membrane surface and release their contents, these um, um, neurotransmitter chemicals. Those neurotransmitters then have to diffuse across the synaptic cleft, and that's subject to diffusion. So that's a relatively slow process versus the electrical synapse, which is relatively fast. And then they bind to receptors on the postsynaptic membranes, and then those receptors are going to do whatever they do. Commonly, they would generate another action potential, but it could be that they cause a change in protein expression as a result of that synapse of arriving or whatever. Okay, because there's a lot of different kinds of neurotransmitters and a lot of different kinds of postsynaptic membrane receptors that bind to those neurotransmitters, you have a large variety in the response that can result from a chemical synapse. Okay, here's an H5P question for you that touches on the last podcast. Okay, so zooming in here, we have synaptic vesicles that are near the presynaptic cell membrane, presynaptic terminal, where the synapse is going to happen. And they will rapidly fuse with the membrane and release their contents, and something happens in the postsynaptic membrane receptors for those neurotransmitters. And here is a electron micrograph showing these synaptic vesicles in close proximity to the synaptic cleft, which is just the distance between these two membranes right here. We see my cursor going back and forth. So it's not a great distance, but it is a distance that you're relying on diffusion for. So lots of options in the postsynaptic cell. What can happen? It could be that the neurotransmitter activates a ion channel to open in the postsynaptic cell, and that ion channel reacts directly to the neurotransmitter. Or it could be the neurotransmitter binds to a separate receptor, which then separately interacts with an ion channel. So a neurotransmitter could cause a postsynaptic cell to fire off an action potential, or it could actually inhibit an action potential. So here is a list of some different kinds of neurotransmitters. You've probably heard of things like acetylcholine, norepinephrine, dopamine, glutamate. So this is just an amino acid that can act as a neurotransmitter. Same with glycine. The monosodium glutamate, a food additive that gives sort of an umami flavor. Some people react strongly to it. I react strongly to it. And it makes sense because it's a neurotransmitter that you're putting into your food. There are even gases that can be neurotransmitters, like nitric oxide. Okay, so for a chemical synapse, let's go over this in more detail. So an action potential arrives at the membrane. How did it get to the membrane? Well, it's traveling down this membrane because of it's regenerated each time at these nodes of RANVA, which are the parts of the axon that are not covered by myelin, and the sodium that rushes in at one node of RANVA travels along the myelinated part of the axon. It doesn't leak out and therefore causes a slight depolarization at the next node of RANVA, making those sodium channels more likely to open, and you fire off another action potential and so on. So that action potential arrives from the cell body to the end of the axon. Okay, so that means that this membrane here that is covering the end of the axon has changed voltage across that axon. That's called depolarization. There are voltage-gated calcium channels, okay, that respond to that depolarization by opening up. And calcium is high outside the cell, low inside the cell. So when they open up with this change in voltage, calcium rushes in to the cell. And the local increase in calcium is going to induce the secretion of some neurotransmitters. So there are parts of the cytoskeleton that will cause these synaptic vesicles to rapidly move to the surface of the cell membrane and release their contents. And this is a complex process here, but it depends on calcium. So as calcium rushes in, it sets that process in motion. Okay. If calcium continues to rush in, even more neurotransmitter is released. So this is another aspect of how chemical synapses can be different from electrical synapses, in that you can get a little bit of neurotransmitter released or a lot of neurotransmitter released, depending on how long the signal persists. And then that neurotransmitter has got to move across the synaptic cleft and bind to a postsynaptic cell according to receptors that are going to receive this neurotransmitter, and then something happens. Okay, something happens, meaning you open up an ion channel in this postsynaptic cell. You have some kind of intracellular signal that tells the nucleus, now let's make this new transcript that we weren't making before. In the case of muscle, you set off another cascade of events that are just going to cause actin and myosin filaments to slide on each other and the muscles to contract. Something is going to happen. Okay, and then in the postsynaptic cell, at this neurotransmitter, at the synaptic cleft, not shown on this diagram, but there will be active mechanisms to recover the neurotransmitter that got released. So you release this neurotransmitter and you can pump it back into the presynaptic cell so you can do it again. And that can happen after these events have happened during recovery. Just some of the things I mentioned before, in that these synaptic vesicles are on elements of the cytoskeletal network that are responsive to calcium, and that's how they can move rapidly to the surface of the presynaptic cell and be released. There are calcium channels in close proximity to this presynaptic cell, and as calcium floods in, it'll cause these vesicles to dock and release their contents, the neurotransmitter. This vesicle docking is dependent on a snare and snap system. So we talked a little bit about this when we talked about the endomembrane system, and that you've got these vesicles floating around in the cell. How do the vesicles know where they're supposed to go? Well, there are snares on the vesicle side and snaps on the receptor side, on the receiving membrane side, and they have to be compatible with each other in order for the membranes to fuse and release their neurotransmitter. There is a botulinum toxin made by a microorganism that cleaves the snare proteins and prevents this sort of thing from happening, from the fusion and release of neurotransmitter. So it can stop neurotransmission, and it can do that for a long period of time, months at a time. This is how by selecting different nerves that will cause certain muscle groups to tense up and other muscle groups to relax, you can change the appearance of facial muscles and make someone's wrinkles lessen or disappear. There's also use of Botox for people with chronic migraines, in that usually involves or can often involve um, tension of certain muscles that surround the head. And so if you release that tension using Botox for a long period of time, then you can have relief from those headaches. So these sorts of diagrams, for simplicity, are shown as a single axon terminal reacting with a single postsynaptic cell. And of course, that's not reality. In reality, you have all kinds of synapses among neurons. And those synapses could be excitatory, meaning that they're more likely going to stimulate an action potential, like those in this diagram shown in green, or they could be inhibitory, meaning they're less likely to fire off an action potential. So how would a cell be inhibitory? Well, maybe you open up a potassium channel instead of a sodium channel in a postsynaptic cell, and you hyperpolarize that cell, making it even harder to depolarize, and therefore it's inhibitory. Here is a cell body of a neuron and its dendrites, and you can see in this picture of in yellow are all one type of synapse that is being made, either excitatory or inhibitory. In red are all the other type of synapses that are being made on these dendrites, either excitatory or inhibitory. And there are thousands and thousands of connections made among neurons. So it's not just one neuron connects with one other neuron. One neuron will connect with thousands of other neurons. And this is where we get some emergent properties of nerve tissue, like the brain and being able to think and form memories and that sort of thing, is that neurons can integrate their signal from and multiple signals at the same time. So let's say in a sort of simple example, let's say you got 10 inhibitory signals coming in and 15 stimulatory signals coming in, then your overall response is probably going to be stimulatory, because stimulatory outweighs inhibitory. Okay, if you get 10 inhibitory and 10 stimulatory, you're not going to do anything because you're equally inhibited or stimulated. So neurons in this way can count how many signals are coming in and the rate at which they're coming in, and then quote unquote make a decision about, am I going to fire off an action potential or not, depending on the different stimuli that I'm getting. They can also keep track of how often am I getting a signal. This is called tone. What is the rate at which I'm getting signals, and there's information in that too. So tone has a big deal to do with the expression of which kind of calcium channels you're going to express in your muscle cells, which has to do with, is this going to be a fast twitch muscle cell or a slow twitch muscle cell? So is this a muscle cell that is better for a marathon runner, or is this a muscle cell better for a sprinter, for example? And that is related to how often am I getting impulses, and at what rate are they arriving at this muscle cell. Okay. Another thing they can do is, it could be that a sub-threshold signal arrives on the cell. So a signal arrives on the postsynaptic cell that says open up a sodium channel, but you're not opening up enough sodium channels to actually reach the threshold to fire off an action potential. But the cell can count those too and say I get a little bit toward depolarization with this signal, and then another signal comes and I get a little bit more toward depolarization, and then another signal comes and I get a little bit more toward depolarization, and then finally the fourth signal comes in and then I fire off an action potential. This would be the equivalent of how loud does something have to be before you hear it? How bright does something have to be before you see it? You don't respond to all signals, you respond to a certain threshold. And this is how neurons can respond to that threshold, is by building up ion concentration over time until it reaches the threshold for the action potential to be fired off.